Okay, welcome to everybody who might be joining this live stream. So we are going to continue our kind of working this session about molecular computing, and we are happy to have some visitors here from Emerald Cloud Lab, who I'm hoping are going to help us understand more clearly what we're doing, and then get us set up so that I can actually, for the first time in like nearly 50 years, do a chemistry experiment again. So let's see, some background here. So what the background here is we're trying to understand what we are sort of calling subchemistry, and that has to do with when you are thinking about chemistry. Usually, you might think about some reaction network, and this is a reaction network done more or less. Let's see, how is this being done? This is being done. Is this using our molecule modify type stuff to actually change these molecules, or how is that working?
Yes, it's using the new stuff coming out in the next release for doing chemical reactions, doing reactions on molecules.
So it's making transformations. It's treating these molecules as graphs and essentially doing graph transformations on these graphs.
Yes. Yes.
And so this is a standard sort of template reaction, and you're running it starting from that initial condition.
Yes.
And in each case here, so these events represent reactions, and this is saying that two copies of that molecule interacting together can produce a variety of other things.
Correct. Yes, and that same molecule that's at the root is available for every single event. There's always an arrow going to each event from that root molecule. So right. It's simulating, starting with a solute, just that molecule. It's present, whatever it produces, it can also react with the products.
Right. But so the thing that we're wondering about is, in normal chemistry, what you think about is there is a total concentration of such and such a kind of molecule, and that's the thing that you study with differential equations or whatever else. What we're interested in is to go below the kind of thermodynamic level, below the level of kind of molecular chaos, where you just say we have a certain concentration of molecules and there's a certain probability of interaction, to the level where we're actually thinking about this molecule, some particular molecule here might have produced this thing, and then that, there's a kind of a lineage of particular molecules rather than just an overall concentration. And kind of the concept there is that when it comes to molecular biology, that there is, this idea of just everything floating around and it's all just a random collection of molecules might not be the right story. And in fact, one already knows that that's not the right story, because molecular biology goes to a lot of trouble to actually sort of bring individual molecules from place to place. That's sort of the introduction. Anybody have any comments or at that stage? I mean, and I think James had a whole discussion about ribosomes and viewing them as an example of the story. You want to say something about that, or somebody else want to jump in here?
Well, okay. We don't necessarily need to belabor the ribosome example here, but just as a general comment, so we're trying to model chemistry using this method that we're calling multi-computation. And in this case, when applied to chemistry, we're doing this thing that Stephen described called subchemistry. But the idea here is that molecular reactions, which at the molecular level are some sort of collision, really, these can be treated as their own individual computational rules. It's like some algorithm, some discrete process that follows certain definitive behaviors. But rather than just study them one by one, we can try and model overall systems that include different kinds of these interactions. And so in doing so, we're presenting as what Stephen called a lineage that that actually visualizes, the different histories in which this could occur. But one reason why we're interested in this is because we suspect, and it's not just the case in chemistry, but with many of these kinds of modeling exercises that we're doing, that when you start to examine models that include, different kinds of interacting rules, that there are some kinds of aggregate or bulk properties that arise from them, which in the case of chemistries could be something stoichiometric, or in biology, it could be some sort of byproduct from underlying processes. In the case of the ribosome, something that we were just entertaining, this week, is that we do suspect that, especially in biological systems, there are certain kinds of mechanisms that allow the kind of like coordination or the synchronization of different parallel processes that are occurring that allow one to kind of capture the computational work that's present in that system to gain new products from them. So in the case of the ribosome, you have these processes of messenger RNA and transfer RNA, and the ribosome serves as this kind of a mechanism by which you're coordinating those two, in this case with codon, anticodon kind of pairings, and you're able to actually, do protein translation and get these peptide chains that arise from them. So that's kind of an overview of the kind of thing that we're doing with this multi-computational, multi-way threaded approach to chemistry.
Right. So, I mean, one of the questions is, normally when you look at chemistry and you zoom out, you say all that matters is the total concentration of molecules. Question is that in fact the correct story? Or are there other kind of nano-perceivable properties of collections of molecules that go beyond just what is the overall concentration? Another way to think about this, another sort of slice of what we're thinking about, is normally when you think about a computation, you think about you've got input, type in 2 + 2, it goes crunch, 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 and produces output 4. The question is that the only possible way of thinking about sort of how you get results from computations? Clearly, in our typical use of computers, a lot of the action is in the ongoing process of what the computer is doing, not what it does when it halts. And so a question in chemistry would be the same kind of thing. To what extent is there, if you're doing molecular computing, for example, is the result of the computation something to do with its dynamic process of going on, rather than, we got this, this pile of molecules at the end, so to speak. And so my kind of, one of my exercise questions is, can we make and maybe this is Bob trying to do this, I'm not sure. Can we make a chemical reaction network where, for example, we generate molecules with a prime number of units, and then it's a little bit like quantum computing, where it may be that sort of in the middle of this big network, we've successfully got a bunch of molecules with prime numbers of units or something. But then the problem is, can we measure that? Can we actually, separate out the things that are just the molecules with prime numbers of units and not get entangled with all the other different kinds of results that we could get? So let's see, just to kind of structure this a little bit. So Bob, if you want to talk more about what you have here, and then Brian, I was curious, you had sent me mail a little while ago mentioning Sidney Brenner and his, he pushed both of us to think more about computational biology. And I regret that I never had the opportunity to do that while Sidney was around and able to talk to us, so to speak. And so I'm sort of curious about that angle too. And then I want to kind of jump in and try and see whether we can understand a little bit about how to actually use Emerald to go beyond just doing sort of computer experiments, and see whether we can actually do some actual chemistry experiments to correspond to these kinds of things. So Bob, did you want to say more about what all of these interesting pictures are?
Well, I would just made a proposal of about half a dozen different Diels-Alder type reaction systems that could be done. Simple ones. The molecules that have a yellow background are known molecules. They're present in the PubChem database. So there is some hope that there's feasibility for them. Probably some of these.
I mean, but we have no idea what the rates of these reactions are.
No, we don't. I do have a little bit of information about reaction conditions for some of the starting materials, for the first step or two. Some of them happen at room temperature, some at 180 degrees Celsius, for example. So they're certainly feasible, I would think, in a typical laboratory. And then also, because we know which ones have been made, we know how far down the chain some of these reactions will go. This one here is fairly shallow, but the one with cyclopentadiene can go quite far. That was one of the smaller ones further down in the notebook. Further up, I mean. Yeah.
Can we ask a really grounding question here on these reaction networks? So I was looking at these in the previous lectures too. It's fascinating. Are we thinking that this is one big, like one pot reaction?
Yes.
Or are we isolating and purifying between each step?
No, this would be a one pot. And of course, you'd be limited by the kinetics of it as to what you would see at any given time point. There are a number of ways you could do it. You could draw aliquots at given times, quench them by cooling them off, and then analyzing the products, or just let it go and see what happens. I stayed away from systems like Wagner-Mervine rearrangements because they tend to go fairly quickly to an equilibrium that usually is fairly deep energetically, and you end up with just one product.
Okay, and then so when you're doing this network discovery and plotting it out here, the starting materials are still present then in the future ones when it's trying to figure out, because everything's independent of concentration.
Yeah, everything here is just strictly, can these two react? If these two react, what do they produce?
Okay, and Brian, everybody's still in the graph, even
right. So what you're seeing is these are tokens, these are events. So that token can be used in this event down here.
Mm-hmm. And it would be used with the yellow molecule just to the left of it and up a little, and it would produce the one to the right and below.
So that's the reaction. It's this guy plus this guy giving that one.
Mm-hmm.
And I mean,
and these would be one pot.
To make a connection with other kinds of things, if this was like more sort of Petri net style, you might imagine sort of using up molecules here, and rather than something like this where you're seeing there is a sort of, this is showing what the tokens can in principle do. This is not showing the using up of those tokens in this reaction so that there's no more of it to go on and do something else. Does that make sense? Go ahead.
I would just say the difficulty of doing that's sort of like a prebiotic soup type experiment.
Yep.
And the difficulty of that, I think someone pointed out on a previous stream, is that the rate constants for those reactions are going to be 5-10 orders of magnitude variant. And they're all second order, right? So they're all dependent on concentration of species as well. So that's why, like, when we're chemists, we don't tend to make huge amounts of the goo here, sort of at the bottom, and do only 1 or 2 reactions at a time, is because most things are being driven by really high concentrations with a rate constant that's going to be much higher than everything else that's happening in the soup.
Your basic point is there are a lot of exponentials involved in these, for example, activation barriers and things like this. And that as soon as there's exponentials around, you're not going to get something that has a large number of different kinds of things in the soup. In other words, if the probabilities for these interactions exponentially differ from each other, what's going to happen here is it's going to prune very quickly into one particular pathway.
Yeah, that's why. And then someone mentioned on a later stream, if you want to do something that's intentionally combinatorial, you work really hard to use reactions that are going to have similar rate constants for all the possible pathways they want to go down in order to get good distribution of mass between that. If you have one primary reaction going on, it's going to massively dominate and take up, so that all the other products are going to come at 0.001% of the downstream.
Right.
That gets even harder if you're doing, subsequent reactions, because those are then exponentially less likely to happen, et cetera.
So what's a good basis, chemical basis, so to speak, for things where there are lots of reactions of roughly equal probability? I mean, DNA is obviously, and things around DNA obviously are one example, but are there any other examples?
Yeah, there's two areas to look at there, I think, that are interesting. Might be relevant here. One is definitely in the combinatorial chemistry space, where they intentionally use particularly high yield reactions, and the diversity comes in the structure of the material that you're adding, but it's the same reaction. So you might add 10 different components that could combinatorially combine differently, and then you do like a split pool for each subsequent set of reactions that you're going to do, but they're all being driven by the same reactive chemistry at the end of the day. There's some active group that's going to connect at the end of the day that has similar rates for all that, as opposed to something sort of structurally wildly different there. Biology does something similar too, right? Where it's like the same linkage everywhere, and the diversity is basically in the sequence of the things that you're connecting, which again connects to structure, but not the actual linkage chemistry between it. And those two both can be controlled in a lab. We do that pretty regularly.
So wait a minute. Let's take the first thing you mentioned, the combinatorial chemistry case. What kind of chemicals are we talking about here?
Oh, that's a good question. Stephen, do you know, Roybosch, do you know of any like good examples to start people off with to explain a typical combinatorial chemistry system? Anything jump to mind? People doing like click reactions and stuff like that.
Click reactions is a good thought.
What are those? You said click or quick?
Click.
Like,
First,
yeah.
it's a
Clicks are like
3+2 cycle addition.
Yeah, they're really high yield and they're kind of low energy, so you can get them to run really readily in a lab.
So what type of molecule? I mean, so what's
It's one side is in a Z, and the other side is a double bond, I believe, maybe a triple bond. I actually don't remember.
anybody know a specific molecule that we can just so we can see it?
Typical click reaction.
See, I still don't have my intuition tuned up at all for chemistry. So I never know when somebody says there's this kind of reaction. It's like, does every chemist here know what that means, or is it like segmented deeply? No. Okay.
Yeah, I was just getting out of medicinal chemistry when this was discovered or developed.
Kind of the important thing about the combinatorial thing here is the reaction is somewhat secondary to the diversity you're generating anyway. So every you've got two totally different sets of structures that could connect, where they have, just think of like one reactive end, and then there's say 5 different variants on one side, and it has another reactive end that you're going to do a subsequent reaction on.
Picture here. Is this?
Yeah.
So this is. Wait a minute. So these are like two different possibilities for how this. What
So I think you're just
R1?
looking at it. You're just looking at a click reaction there with an azide and alkane, and then they're going to the product. There's that happens to have 2 different answers of what could come up.
But isn't that
not really
okay?
In the combinatorial sense, it's like the R1 that would be changing. So you would put a whole bunch of different groups there, and the R2 would be different. And so now you have all combination of R1s and R2s connected to each other. And then presumably, if you set it up correctly, you've got further functional groups on R2, so you can do that again with another set of diversity. You can do that again with another set of diversity. So you might end up running 5-10 reactions in series. And because it's like, n to the 5th power, you get a huge diversity of structures coming out of it at the end of the day. And they're all using the same reaction with each step. So therefore similar rates and therefore similar distribution of masses across the board. That's the general idea with combinatorial chemistry.
Yeah, and you can do this in multi-well plates, and it's completely handled by robots.
Well, it should be in the Emerald case, right? That's the theory.
Yeah, in our case, you could do that, but
yeah,
and then they were using this back in the 90s and the aughts to do exactly what you guys were describing earlier on. There's just trying to look at different shape space to find drugs. So they're trying to bind to some protein to shut it down, and they want something that, usually they would just screen from a whole bunch of compounds that they pulled out of nature. That's like the traditional way to do it. And in this case, they say, all right, what if we just combinatorially generate something that's an extremely diverse set of potential compounds that might be a drug that we could screen for? And then they look to see which of them are going to bind to their protein specifically, fish them out in these big screening experiments.
So when they got that shape space distribution, how did they? So first of all, these were random. I mean, these were molecules with just carbon and hydrogen and oxygen and things like that. They were not anything more exotic than that. Is that true?
That's and commercially available as well.
But wait a minute. But Brian is talking about the combinatorial combination of many such inputs leading to molecule.
The pieces are commercially available. Each of the small pieces that get linked together. So it's very easy to carry it out, and from a dozen of this and a dozen of that and a dozen of something else, you can generate thousands.
Right, but so the thing I'm trying to understand is, in this picture here, the comment is being made, oh, most of these reactions will have exponentially different, rates than other ones.
Mm-hmm.
What is it that makes something if I were just to look at pictures of the molecules or pictures of the reactions, what would make one reaction be a kind of a level playing field reaction, suitable for combinatorial chemistry, as opposed to a reaction where there will be exponential differences in rates?
Oh, that's $1 billion question to ask based on the pure structure, whether the reaction is a good one. Most of it's just done empirically. What we do in synthesis laboratories is we have a rough idea of common types of reactions, and then we try them out and we get rate constants extracted from all of them. And sometimes they surprise us the extent to which they work, but there isn't a hard theory you can point to where you have like certainty on what those numbers should be entirely theoretically.
But so this click reactions thing is something that was found empirically to be something that led to many reactions that would be of roughly comparable rate constants.
Yeah, and what we do is we looked at what's usually called an active group, so that we ignore the whole rest of the molecule and we say, oh, this bit here, that's where we say like there's an R group, and what is on that R group? Not important for the reaction, right? We say the main thing that matters is just that this piece of chemistry here is what's doing the interaction, and the hope is that whatever's going on in that R group isn't going to interfere with it by sterically getting in the way or causing some electronic distribution to be different along the molecule, that kind of thing.
But so the active pieces, what are the active pieces? I mean, is there some well-known piece of chemistry that is the thing that you use to link molecules together to make bigger molecules and things? And
there's a great book called like Named Reactions that you can go for, which is like all the things that the chemists memorize that are like the known active groups on how they interact with each other.
Yes.
Yeah, it's a great book.
Right. I mean, it's to me, it's a bunch of graph patterns. I mean, that's the only thing I can get out of it. Is it's a bunch of patterns that say, if you have a graph that looks like this, which is a molecule, then it transforms into that. But if I look through that book, clearly, if one's trying to do, let's say, molecular computing kinds of things, one's going to have to pick some particular, type of molecule or whatever to use as the basis for doing that. I mean, just as, in ordinary computing, you've got to pick silicon rather than gallium arsenide or whatever else. You have to pick some technology basis. And so and clearly one technology basis that, has been delivered to us is the whole one around DNA and so on. But I'm just sort of curious, the thing I sort of keeping on asking for is there some other kind of technology stack of molecules that people care about for some other reason, where they've been able, in this combinatorial chemistry thing, that was not wildly successful, is my impression. Is that a fair statement?
Yes. But the reasons why it wouldn't necessarily apply to your goals here. So there's lots of debate about whether or not why it was less successful ultimately in terms of number of approved drugs compared to, say, screening natural products. But most of the arguments I've heard around why that's true don't have to do with necessarily getting like a high affinity binder. So solving the problem of do I explore space shape such that I found a molecule that ended up binding to the thing I wanted. But that when you're dealing with natural products, they all have this history of evolution behind them that are selecting against things that just, outright kill biology or can't be digested, or will blow your kidneys up, like all these other things that are going to happen downstream, that the combinatorial chemistry stuff was failing at a much higher rate in sort of those late stage things. So yeah, it was binding the protein you wanted, but there's no hope of turning this into medicine because of all these side effects. Whereas if you work with the natural product stuff to start with, it's already been pre-selected in a major sense. You can't be like fundamentally against any biology because it came from something living to begin with, kind of thing.
But so I mean, okay, the binding question is mostly a question of shapes of molecules. Is that true? I mean, the
absolutely.
I mean shapes and electron density.
Right.
Yeah.
So
that's what we're saying.
Physical shape and electron distribution. Electronic distribution. But so, okay, here's just a naive kind of question. But I mean, is there a characterization? So you've got this space of molecular shapes. How do people characterize that space?
That's a great question. We go look up some Stuart Schreiber papers on diversity-oriented synthesis and read up on that.
Okay, but I mean, back millions of years ago, I happened to be involved in characterizing the shapes of events in particle physics experiments. There was a whole theory that I developed about using spherical harmonics and things to, characterize, to have this sort of generalized characterization of shapes, and it, for that particular purpose. But I kind of would, it is, there's definite things one can imagine people using. I just wondered whether there was a standard technology that people use to say, if you want to look at the distribution and shape space of things. I mean, people talk qualitatively about shape space, and I just wondered whether there was something well known that was quantitative. But okay, but then your statement is for biology, and I'm just as a general knowledge question, I'm sort of curious. For biology, the claim you're making is that a randomly generated molecule will have, is it something that is different from the actual piece that is binding, but some random tail of the molecule would have some nasty anti-biological effect? Is that the typical thing, or was it something related to the actual area where the binding was occurring?
Yeah, I guess I'm making the argument that the failures of combinatorial chemistry are not necessarily relevant to the situation of molecular computing, because you're not presumably trying to, consume these as medicines.
You can't eat them.
Eat them.
Yes.
Exactly.
Don't eat your computer. Fair enough. But so, okay. So, but then in the sort of folklore of combinatorial chemistry, is it like, oh yes, this particular kind of chemical was a good thing to try and combinatorialize, so to speak, or did that either never really emerge, or is that something that's still deeply proprietary, or what's the story around that? I mean, you were mentioning these click reactions, but is there another? I'm just sort of wondering if you're going to pick let's look at this, let's try and run that in the Emerald lab, for example, some particular kind of chemistry, what would be the thing to pick?
Yeah, there's definitely, we would just go look up some combinatorial chemistry papers and see what the most common, there's definitely things that are favored in terms of like good reactions to that. And they all tend to be stuff that is high yield, low energy, so you can do it in a lab, and then with commercially available materials, exactly the kind of thing that makes it readily usable in the lab.
Right.
So there's certainly like review papers one would want to look up in that space and say, what are the most common reactions people use there? And the interesting thing there is that it's different than what we were talking about at the beginning here. In the graph that's showing on the screen right now, the diversity is coming from the reactivity, as opposed to in combinatorial chemistry, the diversity is not coming from the reactivity. It's coming from the structures that are being reacted, right? Like you're using common linkages for each step of the combinatorics. It's the space of the, those R groups, the ones that are not participating in the reaction, but are structurally relevant to shape space at the end of the day. That's where the diversity is coming in.
So, I mean, I'm just pulling up a random paper here, just as an example. This, and this is, I don't know whether this is relevant, but I can't read these things quickly. uh, this claim to be generating some kind of shape space. Hard to know what it's doing here. Why has it got a picture of a, well, I don't know how to read chemistry papers, so well, it's always a mistake to actually pull up an actual paper because you never know what you're going to get. It's probably brilliant, but unreadable by me. But so wait a second. I haven't quite understood, Brian, what you were saying there. So you're saying, in our case here, we are in a given reaction, we are combining this molecule with this one to get this molecule, for example. And you're saying that somehow in traditional combinatorial chemistry, there's a different motif for what's happening.
Yeah, I would kind of break it down as like these look more like prebiotic soup type reactions going on. Like a good one is to look at there's some really interesting classic ones like the Miller-Urey experiment.
Right.
Where they just start with like
methane, ammonia, and water.
Exactly. Yeah. Spitfire and water. And then they let it cook for a long time where it is just going under a soup of all kinds of reactivity. And interestingly, a lot of building blocks of life like emerge from that. The amino acids start showing up and that sort of thing as the sort of downstream products that end up being stable in that network of crazy reactivity that's going on.
Can we, I mean, in our token event graphs, could we make a Miller-Urey experiment out of this?
That would be so cool.
Possibly. The thing is, they were using electric shocks to simulate lightning. And they may have also included ultraviolet light radiation because there was no ozone to protect the early oceans from the sun.
Right, but I mean that's just the weird electric shock is presumably representable by one of our chemical reaction, symbolic chemical reaction.
In principle, but I really don't know what they are. It's almost like blowing stuff apart, and you've got a lot of free radicals that can just hook up, in different ways, in a phenomenal number of different ways. And some things are stable and hang around longer, and
well, so the electric shock, what does it do? Does it just break the molecule randomly at different bonds in the molecule, or what?
More or less. Would that be right, Brian?
No, you're a little, that's some high energy chemistry.
I mean, it just adds a lot of energy, which can make
I mean, it's like what happens.
Weird things.
All kinds of weird things that don't happen in the lab happen, at high energies.
like the early mass spec that were electron ionization instead of chemical ionization, which is much milder.
But as a rough approximation, could you just zap random bonds, break the molecule apart, and see what happens?
And let them glue back together. Yeah, I mean, we could try that. It would be interesting to see what would happen that way.
Okay, but in that experiment, again, this claim about their exponentially different rate constants, and therefore you don't get very far. That experiment again seems to say that there are. I mean, this is what we should be able to study from these networks, right? Because if it is the case that in most cases, when we start filling in the probabilities for these events, that these networks will prune down to something where there's very little bushiness left, then so be it. But it seems like that doesn't always happen. So there may be some underlying chemistry characterization of when the bushiness is pruned versus when it isn't. And that seems like that's a thing to do with the actual details of chemistry. I mean, I think the thing that one of the things we're interested in is assume that the bushiness is not pruned. Then a traditional chemical lab, and I don't know, analytical chemistry kind of setup where you say, and what is the product of this chemical reaction, might not be the right thing to be looking at, because the quotes product of the chemical reaction is this whole range of different molecules. And it might be that there are different kinds of things that you might want to be analyzing about the sort of the chemical system than just what is the concentration of this particular product. I mean, I'm curious whether there's anything that this idea of, observing something other than just the concentration of a particular product, whether that is reminiscent of anything in what people normally do in chemistry? Or is it always just what's the concentration of this thing?
Well, they were going all the way to that email I sent you. I mean, the general assumption in most, even in systems biology stuff, is that all of those reactions are second order. They're all statistically completely independent events. So the molecule doesn't understand the history of how it got there, right? It's just a full distribution every time. And so, yeah, you do end up only caring about like what is the state of the system right now is just the concentration of all the species. And you don't have to care about anything beyond that in order to predict reactivity for the next step of what happens. And that's typically how that's definitely how chemists think about things, because that's surely true with all the reactions running. They're all very high energy compared to what happens in biology. And it's also one pot. So like every time we do stuff, it has things like you're mixing your beaker as much as possible, right? Because you want to assume that everything is at uniform concentrations and there isn't a distribution of concentrations there. You don't have any compartmentalization going on like you in biology. It's a very different thing.
Well, but so the question, maybe Brian, do you want to say more about this email?
The mystery that was brought up. Yeah. Well, I mean that's a whole rabbit hole we can go down. Of saying that, like, so the typical systems biology way to treat things is taking what we do know that is well behaved in chemistry, right? We can sit down with two reactants and there's some second order reaction going on to get to some product, and you can model that using basically transition state theory and say, okay, let's extract some rate constants from this because we know the rate is proportional to some rate constant times the product of the concentration of each of the reactants, and then just solve all the ODEs, right? And like you said, and that's for synthetic chemistry, that 100% works. That's exactly what we do to figure out what's going to happen in the system. I think the argument being made there that might not be totally appropriate, and it's pretty heretical, which is why it was kind of an offhanded statement for Sidney to make there, was saying that I don't know that math is appropriate for the biological systems, because you move into systems biology, the kind of, and I'm not a systems biologist, so I don't want to like misrepresent this field, but from my outside view of it, the feeling is like the best you can get in terms of resolution of modeling what's happening in those cells is to have a giant network of these reactions that are going on that you understand, and to sit there and solve all the ODEs, and that's what you need to predict future events. Now, we came across some pretty basic, this was in the context of Emerald Therapeutics, so I'm glad Catherine is here, but we came across some basic reactivity, even with just a couple of fragments of DNA binding to each other and then displacing each other, where you can get it to pretty quickly violate the math, where it should just be able to like, oh, let's just throw a rate constant on this and extract it out, and there shouldn't be any hysteresis going on, and there's no statistical, you can assume that every reaction is statistically independent. All the kind of normal things that we do in chemistry should work, but the numbers don't work out in a nice way. And we struggle to like fit them to anything. And that brought me back to this conversation that, we had with Sidney, where he's saying it doesn't work like that. If you have all these interactions where the state of the molecule, you can't really draw it and say like, oh, the protein exists and it's just its concentration. There's a configuration of it, whether it's assembled loosely with a bunch of other proteins. It looks more like mechanics, and you have to care about all these independent events, and there may be different math. I think I've come to appreciate that a little bit more in the context of these DNA reactions we're doing, where one of the assumptions to do all those derivations is to say that the reaction barrier is high and statistically low to cross over. So you'd have to look at like the derivations of transition state theory and stuff like that. The whole, when people draw those diagrams of activation energy for reaction, so you say like, oh, if it goes over this barrier, you have some distribution, and so it's rare for you to cross that barrier to get over it from starting material product. That's kind of the basis of, sitting down and coming up with the derivation to say this is how we model the kinetics of this interaction. that there's a lot of low energy stuff, even in the DNA reactions we're doing, where they're just binding and displacing, that don't look at all like that, where it could go symmetrically across a bunch of tiny little humps that are not at a high activation barrier. And this may mathematically change the picture, where you would have to come up with a different set of mathematics to model how one would do that. And it's way outside of my depth. This is why I was appealing for your help on the statistical mechanics, because I'm approaching this from a chemistry and biology side, going down to the physics, not coming up where you guys are from the quantum.
Right.
But it's cool because we could make a pretty good example system right away in the lab that is something we've come across before, where you can just show that it's definitely not behaving correctly. Like, you try to fit it to all the equations, it does not want to fit.
Well, so let's understand a little bit more detail. I mean, because that seems pretty interesting to me. And it's like, experimental science is like, you do the experiment, and if it doesn't come out the way you thought it was going to come out, then, rather than like ignore it because you can't fit it to the existing model, that's kind of, if Planck had done that with the black body spectrum in 1900, we might not have had quantum mechanics, so to speak. But so what you're saying is, you have something with DNA, with fragments of DNA, and you have reactions going on, and the thing where you are computing the probabilities for reactions based simply on the concentration of these different kinds of fragments doesn't work. Is that the basic claim?
Yeah, you go to set up your ODEs, right? And you do the simple thing where you're going to fit the kinetics of a couple rate constants, and you have to decide what model you're doing. So A plus B goes to what on the other side. You draw that out. Whenever we try to draw that out and fit this one particular reaction, it just does not want to fit. In particular, there's a couple of hints of like why it wouldn't necessarily be a classical reaction, because you can fit like the production of product to, and now Catherine, I'm gonna need your help here, like an observed second order, and that works pretty well. But then if you think of that in terms of consumption of starting material in the same models, it will not fit that at all. Like, there is some intermediate that must matter in the context of our reactivity at the end of the day. But every time we try to model any intermediate in, none of the graphs will fit the ODEs when we try to solve them.
Right, but I mean, that type of description is the story, as you say, of you've got the wrong model, or you've got the wrong. But by the time you're saying,
that's exactly what Sidney was saying. We all have the wrong model, and we should be thinking about this other set of math. I think that's what he's trying to tell us. Yeah.
Right. I mean, I think that what you're saying is, there's some sort of hidden state that is, it's really ODEs, but there's a hidden, transition state or something where, that you have to include, which you can't actually see in the lab, but it is visible in the ODE network. But the other possibility is that, it really matters what's happening to individual molecules, and you cannot say that, that you have to take account of the full history, the full kind of Monte Carlo simulated history of every molecule, rather than just saying this molecule, as soon as you've produced it, it's the same as any other molecule of that type, which is what you would get if you had a nice stirred molecular chaos type piece of statistical mechanics. But so, what you need is the full reaction, subchemistry reaction network, so to speak, which is the history of what happens to every single molecule. And you might say, well, I've got a mole of molecules, there's no chance of being able to figure this out. But the point is, what you do is you look at a much smaller version of that, where you are accounting for the life story of every single molecule. And then, you look at sort of larger scale versions of that, rather than just saying, all we're going to look at is the ODE, so to speak. So in other words, you just imagine, what is the actual reaction of these two individual molecules? What does it look like when it comes out? What state is it in when it comes out? And then that individual molecule, will undergo some future reaction before it has been kind of made to be equivalent to every other molecule, of some, before you've equivalence together the, all molecules coming out of that. Let me give an example. So if you're just doing, I wonder whether we can come up with an example here based on molecules and graphs and so on. I mean, the basic point is the claim would be, for example, the different conformations of one molecule. If you say, well, what molecular species is it? It's got exactly the same, diagram, but its actual detailed physical conformation is different. And that will affect how it will subsequently, how it will subsequently participate in reactions. I mean, I think
for a specific molecule out of the ensemble, yes.
Right. That's the point. I mean, the point is to trace the history of every specific molecule, rather than to average over, rather than just to say, oh, we've got a molecule that is typical of this species of molecule.
Okay, then you're basically multiplying the number of, quote unquote, chemicals present in the system.
Yes, by a huge factor, by a huge physical factor.
Just to look kind of fundamentally, I think what Sydney was saying is a little bit different in the sense that there could be a bunch of these hidden states, right, that you don't know about. That's what you're waiting to determine. I think that's the classical interpretation when things don't fit. Is like, well, you just don't understand the reaction network. You're missing components in the thing. And you're right. It could be conformation of the molecules. It could be all kinds of things.
Yeah, some association of different species together. Yeah,
exactly. Some complexation of stuff that you're not aware of. It's not really evenly mixed. There's some compartmentalization going on that matters. That's the classical interpretation. I would say of like when these things don't fit, that's probably your problem. What's interesting though is that it's all based mathematically on this idea that it's not, because you're not actually tracking trajectories of random molecules bouncing around in solution, right? You're trying to treat it as a big statistical system.
Yes.
in the mechanics, and saying, well, there's just a probability of collisions and these sort of things. And I think what Sydney was trying to get at is that assumption itself is problematic in biology, because that's not really what you have in most cases. Like if you tried to go back to the transition state theory derivation, it has a couple of assumptions that fold in normal biological circumstances. Like you have a bunch of these low energy reactions that are just at the floor. Like they're not low probability. They're happening all the time. You're crossing over the transition state in both directions pretty rapidly all the time. And that screws up a lot of our ability to model it using the classical systems that we use for chemical kinetics. And you might instead look at that with a different body of math, because it's not just a question. He's saying it's not just a question, I think, of assembling all the states and then doing the normal ODEs, but saying, well, even the way in which you construct those ODEs in that reaction network could be thought about differently. What if instead you don't just think about, the current concentration of the species, the thing you were saying at the beginning, but also the previous state that it was in? Maybe there's some hysteresis where it matters that there's traditionally you consider there's no momentum, right? There's no associated probability from one event to another, but maybe that's not true. Maybe in some of the biological systems, it's actually really common that there's associated probability. Somebody's already moving in a direction on a rail, right? It's going to keep moving in that direction on a higher probability than just turn around and go somewhere else.
Right. I mean,
and you use different math is what he's trying to say there.
Right. But I mean, I think this is kind of what I'm also trying to say, which is, treat the molecules individually rather than by their species. Don't equivalence together all molecules of a given species. Now, when you say treat them individually, that could be spatially. That could be that the molecule is in different spatial position, which is something we're completely ignoring in all these pictures. And in ODE setup, you're always ignoring spatial location. You're ignoring the momentum of the molecules. You're ignoring the detailed conformation of the molecules. You're ignoring lots of kinds of things. If you really, I mean, you're, but.
So there are simulations that people do for that called like molecular dynamics, right?
Indeed.
And that's where you're looking at potential energy surfaces, and you can simplify them to like classical mechanics type things, or you can go semi-empirical or straight up as close as you can get to Schrodinger without, melting down the equations in your computer to try to get to that direction. And those are just completely different than kinetic modeling, where it all ends up in a rate constant and it's statistical and you don't care what's happening at the individual level. And so there's this question of, in order to simulate biology, would you need to get down to the level of understanding the molecular mechanics of all the events? Or can you do this picture of it's a soup of reactions in a bunch of different compartments? And if I understand the set of reactions that are going on, the different compartments that are going on, and the trafficking between them, I could use, these statistical models for the whole thing. And I wouldn't need to understand the individual positions.
Right. So let me give an analogy. So I mean, in fluid dynamics, for example, ultimately the fluid is made of a bunch of molecules bouncing around. But we know that under many conditions, on a large scale, you can just use continuum PDEs to represent that fluid. Now, there are conditions in which it matters that the fluid is made of discrete molecules. For example, if you're, at sufficiently high speeds in hypersonic flows and things, you can get situations where, there's a shock that is thinner than the mean free path of the molecules and things. And then it really matters which kinds of molecules you have and exactly how they behave. But much of the time you can aggregate it up and just look at the for depending on what you're interested in. If what you're interested in is the overall velocity field, if that's what you're measuring, then you can aggregate it up and you can just do fluid dynamics. But if you were interested in different kinds of things, if for whatever reason, if for example you're trying to compute with molecules in fluid dynamics, and you notice that if you measure some correlation function between molecules, if you could measure that, then you could discover that you can make computations using that correlation function in very different ways than you can make computations just looking at the overall velocity. Now, in terms of the modeling, like, for example, a thing I invented in the 80s is a way of doing that kind of fluid dynamics modeling using cellular automata, where instead of having realistic molecules at every site, everywhere, you just have discrete, here I pull up a picture. I've got easy to find a picture of this. Yeah, this is kind of a typical picture. That, instead of having realistic molecules, you just have presence or absence of molecules in different directions on a grid. And when you aggregate that up, if all you're interested in is the velocity field, this correctly aggregates up to the standard differential equations. Okay, but if it turns out that, so this is really the question, in this case, we're getting PDEs, not ODEs, different type of setup from chemistry. But the question would be, if in fact, you were a little critter that lived on the corner of this plate, it might be that what matters to you is utterly different from this overall velocity field that you get by aggregating the thing up. So if in biology, what matters is something other than this, total concentration thing, then you can end up with a, different story if you start off with individual molecules. I mean, I think this is sort of the analogy, and maybe we need a better example for chemistry of something like this. I mean, here what we've got is, we've got spatial degrees of freedom and so on, which we don't have at all over here. I mean, does this make sense? So the kind of thing, one of the things that's come out of our physics project is how generally it's possible to kind of aggregate things up if you have an observer of a certain type. That is, if what matters is something where the observer is, a computationally bounded observer with certain characteristics and so on, then you necessarily get these sort of continuum type behaviors. But it could very well be that the observer that matters for biology isn't that kind of continuum-like observer. I mean, there's nothing that says that, and the same thing with molecular computing. If in molecular computing, all you're saying is, I just want to do it like synthetic chemistry, and I just want to see how many of this molecule I produce, that's one thing. But, if there are aspects, if there are ways to essentially measure features of this reaction, of this subchemical reaction network, so to speak. And I'm showing this one, but I don't really mean this one. I mean the one that deals with each individual molecule separately. If there are characteristics of that are observable, then those may have all sorts of interesting features that effectively let you do more sophisticated computations, etc. Did that make any sense? So, I mean, I think that one of the things that's probably a modeling challenge here is we should get more specific about what happens with individual molecules. Okay, any comment from our if we don't deduplicate in this graph, this involves token deduplication, right? If we do not token deduplicate, we will see every individual molecule. True statement, guys?
Yeah, that would be the case. But here I don't think we generate duplicates in this particular example.
But don't we mean species of molecules here? We're not saying individual molecules in the.
It would be species,
soup, right?
yeah.
No, but token deduplication will give you every individual outcome from every individual event. We will not deduplicate. Right here, every time we get a molecule that is of type 6 or whatever it is deduplicated into that.
Right, but there's only one edge going into that, which means it occurs only once in that network.
Right. So this one doesn't have the feature that, in other words, in order to get a given molecule here, there is only one path to get that given molecule.
Exactly. There are some others in this notebook where you can get to the same molecule by different routes.
Right.
Not this one, but further down, when for example, reacting cyclobutadiene with isoprene or cyclohexadiene with isoprene.
Okay.
This one here, for example.
Right. So here there would be many. Okay. I mean, if we say there's an individual molecule, and look, the correct way to model this is there isn't just one copy of this molecule here. There are zillions of copies of this molecule.
Right.
And each copy, which we can which we can do with the same technology here. It's just a question of what we're deduplicating. And right here we're deduplicating both tokens and events. So this is just showing the overall chemistry as opposed to subchemistry picture of this. We could get a subchemistry picture by saying, instead of like here, for example, we're allowing this single token to be used twice in that event. We don't have to do that. We can say, let's break these apart, and if we want 2 copies of the token, we actually have to have 2 copies of the token. Does that make sense?
Mm-hmm.
If you did one for each molecule, though, there's a fundamental shift there in that those orange dots right now, like those are the kinetic ODEs. Going into those equations is the concentration.
Exactly.
Right. We don't really, you have a totally different body of physics to figure out what happens when it's just 2 molecules floating in a vacuum.
Well, but the good news is, hopefully I can just run this here and we could just make one of these. Hopefully. And then I want to really try and look at, okay, what's Bob, is this expected to be this slow?
Oh,
that's coming.
There you go.
Okay, so this is going to be, why is the supply reaction not?
Are you running a prototype? If you're not running the prototype, you need Jason's Packlet.
Well, I mean, in general, it hasn't been merged in yet. It's for 13.1.
Yeah.
Oh, I understand, but I'm running the prototype build. I think I am.
No, it said 13.01 there when you did.
Well, then I messed it up. I messed up completely. Ugh. Okay, hold on.
Yeah.
Hold on. Sorry, this is let's see, Nathaniel, I think you sent through the agenda here, so I need to pick that back up again and move it and put it in 13.1.
It's also in the Dropbox if you want to access it there.
That would be convenient. Let me see here.
Search.
Is it in molecular computation?
Mm-hmm.
And
meetings, live streams.
I don't know. There's nothing called meetings there. Hold on. Oh yes, there we go. Okay, 428. Right. Okay. That's not what I want to do. I want to move it to that. Okay. Finally, here we go. It's this. And which is the one I was going to look at? It doesn't really matter. I'll look at any of these. I'll just look at this one here.
Okay. Well, evaluate the initialization cells.
Yeah, I know. It'll do that.
And you also have to evaluate the Diels-Alder reaction, which is up a little bit just off screen.
There we go.
Yeah.
Okay. So that's the specification. By the way, for the Emerald folk here, we're curious about your reaction to our reaction stuff here.
So
By the way, why does it say pattern
Yeah,
reaction? Why is that still?
I still don't. So it still appears to not quite work.
Okay,
I don't.
Can I load a packlet to get this to work?
I don't have anything ready at the moment.
Okay, so how did you do this, Bob?
I ran yesterday's prototype build.
But this is April 27th, so this is yesterday's. So if I try running this, is it just going to fail?
If it's still blue, yes.
It's failing. Okay. Okay. Well, anyway, so look, the thing to understand here, it's a shame. The thing to understand is we can absolutely not, I mean, those lines there give us token deduplication. I don't know, maybe Nick has an example where we can try some much simpler case of token deduplication just to illustrate how that works. We have some very elementary example of that. I mean, we can just say, wait a minute. So this is, Nick, any examples, any suggestions for a simple example here? It doesn't here. I don't know.
Nick?
Oh, okay. Well, okay. Let's not do this. But for Brian and other people's benefit, I mean, I think, you know, we absolutely can make a version of this where every molecule is separately accounted for. And then, you know, the PetriNet version would be to have some kind of indicator. They call them tokens, which was very confusing because we're calling a different thing tokens. That is sort of a count of the number of instances of that molecule. But, you know, in the most direct case, we're just representing, every node here represents a different, not molecular species, but a different actual molecule. And so that would be the analog of what we're doing here, where we're looking at, instead of looking at the velocity field, for example, we're looking at individual, you know, each individual particle in the fluid. So I think what you're basically saying is, and what we should be doing is looking at, you know, how can we characterize, you know, it's hard enough to deduce the rate constants. It's yet harder to say for these individual molecules that where these molecules have interacted, they've interacted in this way. The thing that's come out is not just a generic molecule of this type. It is a molecule that had this particular history. And so then the question that we have to ask is, I mean, in the case of the fluid, one of the simplifying features is we assume translational invariance. That is, that a collision happening in one place in the fluid is going to work the same as a collision happening in another place in the fluid. But so I think the sort of basic thing we would have to do here is to ask the question, you know, what can we say? How can we not have to compute separately all of these molecular interaction probabilities, which are, you know, far away from what anybody's able to compute right now? You know, can we make some simplifying assumption about those, you know, each different case of these molecules and what their interaction and how they'll interact? So, I mean, but I think, yeah, there's a definite path here for looking at one of these things in a non-aggregated way. And we should just study that. I mean, we haven't studied that. There are much simpler cases than the ones we're seeing here, and that would be a convenient thing to do.
Can I make a suggestion on model system?
Yes.
The problem with doing that with like pure organic chemistry, or like a Diels-Alder reaction, is that those things are incredibly well behaved in the kinetic case. Like if you watch something like React NMR, it's the most satisfying, beautiful things to watch everything fit the ODE in real time as it reacts.
Mm-hmm.
We could give you some model systems of, I know, we've been trying to avoid biology here, but there's some really basic just DNA interaction systems that we work with at Emerald that definitely don't fit those things nicely and would be really amenable to that kind of model that you're saying that's sort of in between
Sounds good.
full molecular mechanics,
okay.
but doesn't exactly fit the statistical models either. It doesn't really work well in that regime.
That's interesting.
I'd be very curious to see that system because I've done a lot with kinetics, various, epidemiology, chemical kinetics. Yeah.
Yeah,
Fair
and
enough.
in
Okay.
chemistry, it's super well behaved. So finding one that violates chemistry, I think they do exist, but they're going to be rare. In biology, I think Sydney's supposition is that they're actually really common, and that's why it's so difficult to try to sit down with just a series of, a net list of all the reactions that are going to happen, and then say what's going to happen in the cell.
But I think Sydney, with infinitely more knowledge of biology and chemistry than I have, has come to the same conclusion that I've come to, namely that it that there's a sub to chemistry, so to speak, that might matter. And I suppose, when you start seeing these things of biological cells that have all these elaborate transporting mechanisms of little fibers and things moving molecules around, it kind of does suggest that there's a little bit more going on than just you stir the pot and everything is equally mixed. But okay, maybe we should turn to Emerald here. I guess I was hoping for some sort of elementary tutorial example, whatever, of what's a good.
Well,
Yeah,
Bob, is there some version of this that we can demonstrate that doesn't involve the cutting edge, so to speak? Say from a notebook that's been saved to Dropbox from previous weeks, or something that we know works.
All of these reaction things are using the stuff that's coming out in the next release.
Right, which appears not to be in the prototype build.
And tragically, we're on. If you version us, we're way behind because we're stuck on CDF.
Yeah. Nathaniel just asked me if I was running desktop or Mathematica, and I'm running Mathematica. If that makes a difference, I wasn't aware of it.
It can do. I mean, it depends on which build was.
Yeah, I only run Mathematica, and I was running yesterday's build, and.
I mean, that's one of the features of having multiple desktop products, is that their prototype builds may not be in sync, which can be a good thing. Let's take a look at this one.
Yeah.
Well, okay. I mean, I can run this. I can try running this guy in that version.
So either template reaction or pattern reaction should be a symbol in the prototype, I would assume. I would also assume that lots of things don't work, but I don't know. It's not testing anything for this.
I ran the whole notebook before I gave it to Nathaniel yesterday. So.
Okay, well, right now this thing is literally not coming up. So that's what you get if you run a prototype build.
Yeah, I know. We're on the bleeding edge.
So I don't know. That's sort of hopeless. It's okay. Let's say, what are we calling it? Pattern reaction?
Yeah. So that looks like it's live because it's black.
No, the kernel was dead. It looked like.
Yes, the kernel is dead.
Oh.
It's much less alive. Well, actually, it's still black. All right, let's try one of your things. Well, look at that. Look at that. There you go. Well, let's see if this is runnable. I have no idea if it will be. Let's see. That'll be exciting if this actually works. Look
Bingo.
at that.
Nice.
Okay. All right, here we go. Here's our molecule. And now your strangely named G is this whole thing here. Okay, so let's take a look at this guy. That's a token event graph. Let's if we just run that token event graph for one step, let's see what happens. Still grinding for ages. There we go. Okay, so that's one. So that's with deduplication, right?
Yeah.
So now what we would do to go to sort of subchemistry is to say, in here, let's see, what are you starting with? What's the initial condition here?
The initial condition is empty because I'm using token decomposition function to inject the starting material at every event, making the isoprene molecule available at every event.
All right, okay. So let's just make let's get rid of this token. Is this the thing I'm looking token?
Yes, that's the one there.
Okay, so if we get rid of that, and then we just put a couple of isoprenes in here.
Yeah.
Okay. Now that does the same thing. So now let's get rid of token deduplication. Oh, come on. What's happening here? Why is that? Well, okay, let's get rid of event deduplication. There we go. Okay, now what are we seeing here? So, but why are we only seeing one copy of that isoprene molecule? Why are we not seeing multiple isoprene molecules there?
I don't know. Ask
Nick.
Nick.
Is Nick with us?
I reached out to him. I haven't heard back.
I mean, he's
several time zones ahead of us.
He's technically on the call, but I haven't heard from him.
Yeah.
Okay. Well, so what I was expecting this to do was to have
instead of token deduplication false, make it none.
Oh, there we go.
There you go. There you go. That was it.
Okay. All right. So this is now still a little confusing. This is because this is, I would expect there's something still fishy. Oh, I see. If I go event deduplication none, then you're saying I'm going to get, no, I'm still not.
No, event. I don't think event deduplication has none as one of the values.
But what I was expecting to see here was this individual molecule, and that.
Yeah, it's doing that, and it's doing tuples. So the molecule A is being used twice by itself. It's being used once with the other, with B, and then B is being used with A, and then B is being used.
So we want to be able to do it where we individually take these molecules, where we don't allow this, a molecular bath of molecules, so to speak, to just pick out another molecule. We just want the individual molecule to be used.
That would be more like a Petri net, I think, then.
No, a Petri net has a count of the number of molecules.
Okay. I haven't used token event graph that way.
Right. So we should be able to do that. But what we should see here is just these two molecules interacting to produce in the different ways that they can react to produce. Oh, I see. Actually, you know what? This is probably right. Except that this doesn't seem right. It should be two different molecules always interacting to produce something, but then they can produce multiple things. And if we don't deduplicate these events, they should show different actual reactions. We should be able to take this apart. We're not doing it correctly here, but we should be able to take this apart into all of the sort of combinatorial possibilities of how those two specific molecules interact. And then what we're doing, and we need to actually look at this explicitly, what we're doing as we go towards ordinary chemistry is we are doing multiple levels of deduplication.
Okay. This is using tuples to select tokens. And so it's going to do 1 and 1, 1 and 2, 2 and 1, 2 and 2.
So that's what this is doing here. So this is duplicate products by. Okay, so what we really need here is instead of something like that, we need
And what's the other option? I think.
Well, just apply reaction. Why isn't it just apply reaction? Isn't that going to give you? What does that do? What does apply reaction? Let's just see what that does.
Okay.
If we apply the reaction to what is the input here? To isoprene. Okay, isoprene to isoprene.
Yeah.
I don't know what the all does. What does the all do?
It to give all possible mappings of the reaction onto the reactants.
Magnificent. So there you've got. Those are the outputs.
Those are the outputs, and some of them.
I don't know whether we need flatten or not. I mean,
we need flatten to get a single list back, because you're getting lists of products for each possible way the reaction could be instantiated with the given inputs. And since we've got multiple instances of the double bonds present.
This is what I was, this is closer. Let's see, is that what I'm expecting or not? But this is closer. We're getting, it's the same thing. We're getting, no, not quite. We're getting a little bit closer here.
Yeah.
I mean, so I'm not quite sure why you can get that individual molecule. I remember there's an option for this, for whether you can have multiple uses of the same thing. What is that option?
I don't remember what it is, but it's whether you use tuples or subsets.
I understand, but we need to look up. Okay, so
let's see.
For the token event graph, we need to know whether let me see, token event graph.
Can I ask a question?
Yeah.
Are we going to be able to feed in, instead of just Diels-Alder, like all of named reactions, and then see the whole potential schematic eventually?
Well, yes. Yes, that's the whole point.
Cool. That would be so cool.
That's the whole point of the supply reaction function. That's coming. It's a. Let's look at what the Diels-Alder thing actually looks like here. Let's just see. Okay, what is this? Our final representation? Oh no, this still has. So that's the pattern variables, right?
It's showing.
No, then that's the correspondence between atoms.
Between reactants and products.
Okay,
where they start and where they end.
This has no pattern variables in it. Is that correct?
No, I wrote it using smarts because it was more compact, and the pattern variable stuff is evolving.
No, but I understand. But it doesn't. To specify this reaction does not require pattern variables. True or not true?
True.
Okay. But so Brian, when you have those R's hanging off things, those are pattern variables for these graphs. And those can also be represented this way. But so, the thing that's happening here is we're literally taking, you can see what's happening. It's just, I assume that what we can do here is if we say molecule plot of that, flatten of this, we should see all those things. Yeah, there we go. Well, why does it label hydrogens on some of them?
Let's see. So there's a set of rules as to which hydrogens get omitted and which ones don't. And so at the end of at a terminal alkene, rather than just have the two lines terminating in with nothing there, we put in the hydrogens.
Okay. So anyway, so yes, Brian, the goal is to have all those name reactions available like this.
That's very, very cool. And then the thing that chemists do is retrosynthetic analysis, right? So sometimes they want to go the other direction too. Feed it in backwards of like product, what could I break it down into?
Right. You see, the thing is that now that we've got this represented in terms of things like token event graphs, we can run it the other way. Theorem proving technology to go find paths and all kinds of things like that. We have no idea what the reaction rates will be, but we can, find the combinatorial paths in very, efficient ways, we hope. Just to give a sense of that, and then I really want to come to actually Emerald and real world chemistry, but
the sync up here, by the way, is I totally want our input to the actual chemical reactor to take this stuff in How cool would that be? You just straight up say, go run this in the lab. I don't know the rates yet. Go find them out.
Yes, exactly. That's what I want to. Right.
That'd be amazing.
Right. I mean, it's just like we have a thing that 3D print. Let's actually run the reaction, so to speak.
Absolutely.
I was describing your technology stack a bit in some completely different meeting, and it was like somebody was saying, so you mean if you say run the reaction, that's a function that might take hours or days to come back? And it's well, yes, but it's actually doing a real thing. It's not a function that's running. It's a physical thing. Anyway, the
time axis moves in real time.
What is the typical time to do? I mean, assuming that you have the ingredients available at your facility, what's the typical sort of running time for things? I mean, is it ever minutes, or is it usually hours, or what is the typical time?
We try to schedule it. If you're giving us something novel, the first thing it's doing is like resource planning in the facility to get access to it. If you're continuing off of a script, so you already did, we can path plan ahead and we know, 5 experiments ahead what you're going to do, then yeah, it's minutes to get through. If it's like you giving me something from scratch, 0 to 60 right now, we try to target it to be 5 hours. So that's generally the time. And that's presuming you have all the materials in hand too. There's a piece here of like, we only have some certain set of starting materials on hand. We don't have the entire catalog of all things available in the universe.
But usually that's a 1-day delivery for materials. Is that true from the standard?
3 to 5, but yeah.
Okay.
something in that.
Is it 3 to 5 because they have to make it, or is it 3 to 5 because of just
depends what you buy, but also their warehouses are kind of ancient and slow. They're not moving it like Amazon schedules.
I see. Just to give a sense, by the way, once this is all the same technology as we're using for all the stuff with mathematics and so on. And this is, these happen to be axiomatic systems and sort of mathematical expressions, but they could as well, exact same technology as we're using here for chemicals. But with mathematical expressions, we kind of already know that we can use automated theorem proving to find, giant paths. In the full network, it can be very hard to find, how you get from here to there, or whether there's a way to get from here to there. But what automated theorem proving does is to do the path finding through that network to see how you would get from the initial condition to the final state in ways where, were you doing this combinatorially, it would be, 10 to the 100 possibilities, but you can do it much more efficiently. Now, of course, when we're doing it with actual molecules, we can afford to have 10 to the 20th, things being done, which we can't expect to do in an ordinary computer, which is why we should care about molecular computing.
Well, but even in the context of planning a synthetic route, that's most of what you do in total synthesis. And I think the state of the art, people are just doing it combinatorially, as far as I know, where like at most sophisticated, do like an A* search type of thing.
Right. Well, product. We should be able to do much better than that. I mean, in other words,
that's already gigantically valuable. That's awesome.
And the thing that's cool, as you can see, is that because this formalism is really the same, we should be able to directly, just use one set of tools in the other case.
Yeah, absolutely.
So, okay, maybe we should turn here. Okay, where do I start? So I've got your command center up here, and what would be just maybe a little bit of a tour here. So I can pick an experiment, and these are all different. Are these different kinds of experiments?
Yeah, everything with the header experiment in the front of it does something physical in the lab. So if it has the prefix experiment,
okay, so sample preparation would be. So what would be like? Let's say I was going to do this.
The most basic thing is like make a stock solution of stuff.
Okay.
It's just like mix a bunch of things together in in a beaker kind of thing. And then you're just going to, if you do that, you can select sort of the objects you're going to put there. I think you want a different overload though than one you picked, because you're looking for one where the formula is already defined.
How do I go back?
In this case, because you just started, I would just exit out and start over. See on the right there, next to the closer. Bottom right.
Here.
Well, that looks like it's CDF
This one
player
here.
vomiting. Yeah, there you go. That's what you actually want.
Okay, all right. Fine.
Resizing is angry. Yeah, go to experiment.
Yep.
And then under stock solution, the overload you probably want is stock solution. The like fourth one down there is the components.
This one here.
Yeah, and there you could just pick a bunch of components. So you do that one. Okay, so now it wants to know how much of what. So you see, it's like amount and then compound.
Right. So by the way, in our this whole quantity instance mechanism, I mean, you must have some analog of the quantity instance mechanism here to represent this.
Unit handling. Yeah, we it's some hybrid of the very old unit system from version 8 and what you guys do, where we use multiplication and up values to convert that into a quantity wrapper. It turns into a quantity wrapper at the end of the day.
Okay.
But you can type in things like 5 space gram, and that still works in Emerald. Capital G, like as a symbol.
Okay.
Yeah, it's okay with that. But in the GUI there, it wants a drop down for the menu. So now that's 5 gram. You're like doubling it up. So just give it a number there.
Okay.
Yeah, it's trying to make it.
Can I avoid the GUI? Can I just type code?
Yeah, absolutely. I mean, you can just go in collapse this and then just start typing out experiment stock solution.
Okay.
And you can pull up the help file for it. It looks like a relatively normal help file. Exit's okay.
How do I get to the help file?
Well, there's a upper right to pop up in the help system. Yeah, there you go.
Oh, great. Okay.
Or just in Notebook, you say experiment stock solution. So yeah, just in the search to experiment stock solution. Yeah, you want the one from our package. There you go.
Oh, I really like your I mean, I've been seeing your online documentation. I mean, the fact that this is all now on the web, these are great pictures, and this is really,
Thanks.
You should
like
make
that drum roller.
Yeah, right. I mean, what it's going to show you here is a crazy exhaustive list of all the instruments you do to mix it. You'd be surprised how many different ways chemists have to mix things. There's a lot of options. So there's a good, 20 or 30 devices that you could potentially mix your stock solution in.
Oh, wow.
You should link this to the web URL for this, even from end product.
You mean get to the problem with that right now is in the CDF. If you try to open URL, because it thinks it's a browser, it half opens it in CDF and blows up the. We really have to get off.
Yeah. You need to get into the true cloud.
Get into the real cloud, and not be on CDF player.
Right.
Actual URLs.
Okay, but if I were to,
I mean, if you just want syntax, just scroll all the way to the bottom. You'll have examples.
If I just take what is this experiment stock solution? If I type into a generic, let me just see.
If you just go to the bottom of the help file, there should be some example commands you can just pull out and start playing around with.
No, I want to show the coolness of the fact that you can just do this. And okay, let's see. Here we go. So this is this function. I just think it's great that this is on the web. I mean, this is people will actually be able to understand. I mean, for me, for example, as trying to understand what on earth chemists do, this is super useful. Okay, so so let's go down here. By the way, you probably should have some jump to, okay. All right, so here we go. Sodium chloride, potassium chloride. Okay. So now here, I want to go over, presumably, where do I go to get?
Just collapse the builder on the right there. There's the double, the chevron, just press that.
Okay.
There you go.
So now I go click to copy, and there I go. Now, what's going to come back? So I run that, and it's just a symbolic object. Is that true?
That should have evaluated. Guys on our end, do you know why that's not evaluating?
Maybe there's a hole around it. Try full forming.
Or is there some nastiness coming from the copy from the web?
Looks pretty good to me.
Looks good to me, yeah. Is the listiness right, Stephen?
Yeah, let's put you on the side, Stephen. Is that the right overload?
Listiness looks okay to me.
It looks, yeah.
Component, solvent, full volume, right?
I'm a little bit puzzled.
Okay.
Oh,
What
model
happened?
sample chemical.
Oh, oof. Oh,
Whoa, that's super
embarrassing.
old.
Oh
How
yeah.
did that happen?
Okay. You've
Extremely
got old
embarrassing.
examples there.
Oh, sorry. Okay, if you just delete the word, sorry, this is embarrassing.
Chemical. The subtype.
Delete the
Yeah,
word
remove
chemical.
chemical,
Yeah,
and
get rid of the
there's
subtypes.
a couple of examples there. How did that
Yeah.
slip through?
How did
Okay,
that
we
slip
made
through
a-
testing?
I don't know. Sorry about that. I'll fix
That's
that
all right.
today.
Yeah, that's
Okay,
an
so what do I do? I get rid of the word chemical in all of these?
Yeah, we removed all the subtyping of the samples because they want them to be general.
Yeah, because you could have a chemical mixed with like a nucleic acid. And then
Yeah,
do
what
you
happens
call
if I
it?
mix it with a cell now? The nightmare of dealing with that. So samples are just samples now.
Okay. Running
That's
with
doing
run.
something. Here we go.
There we
Now,
go.
what's it going to do? Okay. What does that mean? So what's happening here?
Right now, it has to now like simulate the entirety of what happens in the laboratory to make sure this is all going to be okay. It may come up with a bunch of complaints or realize that it has to get some resources you don't have access to. It's checking all the equipment to make sure you're not going to melt it down while you're doing these things. It's making sure that those amounts that you're going to weigh out are going to work out with whatever measuring devices that have to be figured out. And then it will come back to you with a protocol description. So you'll get an object. You'll get an object back in Constellation, which is our data source. So there you go. So now it says, all right, you have a protocol stock solution. Now, if you go to your experiments tab on the top, all the way at the, that one.
Oh, I'm still admiring this. Okay. So this is, so let me just understand. So I've got 80 grams of sodium chloride, 2 grams of potassium chloride.
Mhm.
And this is just combining together and dibasic sodium phosphate, whatever that means. And then some amount of water, a liter of water.
And you're gonna make a liter of it, and you're gonna fill the volume, so you're gonna end up with exactly 1 liter of that solution at the end of the day. So it's gonna put the solids in. It's gonna add enough water to get to 1 liter.
Okay, so now I go to experiments.
Exactly, and now you should have one in your cart because you haven't, purchased it yet. It's sitting there waiting to go.
So what does that mean? I go to cart.
Yeah, go ahead and click on cart. And there it is. So if you click on that guy now, I think you should get a button to accept it. There you go. Confirm protocol. If you do, before you do that, that will legitimately go make it in the lab. So
well, I hope your example is not. I really want to see this.
It'll be safe. You'll be okay to make the thing, but that will go make you that stock solution. A liter of it will show up in virtual inventory for you in the lab. But
virtual, it's not virtual inventory. It's sitting in South San Francisco as an
correct.
actual
In South San Francisco,
water.
there will be a liter of water along with all those things as soon as you hit confirm protocol, and that takes about 5 hours to get into queue. That's what it's doing. It's resource planning to make sure those chemicals will be available, those mixtures can be available for you. The people who have to traffic stuff between the machines and clean them afterwards, all that stuff has to be provisioned.
So so just so I understand this, can I see the things that you computed for me here about what's going to happen? Can I see any more information about that before I?
Absolutely. Go back it will show you the test. Go back to your notebook. In the upper left is the actual notebook itself. It says test 01. Test. Yeah, that guy. Okay. Right. And why is our page not showing here? CDF is unhappy. I think you're still on scratch page 1. Is that where we added it?
Oh no, wait a minute. What's this? This
is some test page from before.
Oh, okay.
Yeah, go to scratch page 01. It's in the tab. You see right next to.
Oh yeah, there we go.
Yeah, that's where we added it. We should probably save that as something if we're actually going to run it.
Why is this not coming up?
Yeah, why is it not coming? What's going on with the player showing the notebooks?
Maybe CDF doesn't perform well if you're in parallels. I think Stephen has this.
Yeah, I'm running this on. I'm running it in parallels. Is it possible for me to see this?
Yeah, can you try clicking in the middle of the screen a bit? Maybe that'll jog the CDF awake. There we go. It looks like it's kind of coming through now.
Try just scrolling up and down.
Okay.
Okay, there we go. Now it's happy.
Okay.
Right. Yeah.
Now,
okay.
How do I see more about what was actually done here?
Copy paste the input line there, and let's put different headers on it. So there's nice headers to like simulate what might happen here. So you could do. What will be interesting is do valid, and then put a Q at the end. So valid experiment stock solution Q, and then throw a flag on that says verbose goes to true. That'll be interesting. Is it still verbose? Did I have the right?
I think it's verbose.
Okay, what this is going to do is show you all those ugly back-end checks we do to make sure you're not going to burn our lab down. So here it's going to go through and check all the composition stuff. That stuff is available. That the precision is okay for what you gave it for the stuff that has to be measured out. Do we know Stephen, how many tests are you expecting to see for like stock solution? I don't usually do a lot of those.
I would expect like
a couple hundred.
Yeah, a few hundred.
Are they going to spit it all at the end, or they? It may take 20 seconds.
It is going to spit out at the end. Yeah, it's gonna.
But so the interaction.
Oh, here we go. Wow. Okay, 1,600.
More than 100.
There's a lot.
Yeah. Okay. So this is. Oh, I see. So it's going to be computing them all, and then it's going to. Yeah, your life would be a lot easier if you were either using our now more streamlined cloud, or if this was running locally on my machine, it will be communicating with your backend object store. Is that true?
Yes.
Do you have
every time you see that icon pop up, it's talking to Constellation. So most of what's slow in that computation is it has to pull, a ton of database information about the lab, about those chemicals.
But it's an SQL database eventually. Is that correct?
Yes.
So we have a lot of now nice. Oh wow.
Yeah, there's a lot we have to check to be sure this is going to be okay. Very pedantic stuff, like that the pattern function is even defined, and then it's going to check. It's a lot of pattern matching, and I don't know. Stephen, you wrote this? What are interesting things to look at in the list of tests?
I mean, a lot of the stuff is boilerplate, like you said. Like all of the options exist, but there's some stuff. You said filter volume goes to volumetric. It's like you can't do certain things in volumetric flasks because they're kind of special containers. So there's checks that make sure, that you're not trying to do something.
Stick an overhead stir in there because it doesn't fit.
Stuff like that.
Checking the precision of the amounts that you specified to make sure that they can be achieved using the instrumentation we have in the lab. Like the tolerance of the balances, or the resolution of the balance can weigh out the precision of what the mass is that you specified. Otherwise, it rounds it and gives you a warning. Stuff
this
like
is
that.
Sort of the hairy guts too. If we do an option, let's do an option call so you can see what it figures out for the resolutions. So if you do, again, copy paste the same thing and...
what happened there? Oh my gosh, I pressed Command L thinking that...
Oh, did we crash it?
No, we got back to a Windows sign-in. Shoot.
Oh, it logged you out of Windows?
Yes. No, help. Okay, could somebody get hold of Roger for me?
Sure.
Roger's in the Zoom.
Oh, well, maybe Roger can tell me where to look for the password for my Windows virtual machine.
We could show you since we have the benefit of having Catherine here. Okay, if you want to see what we're just making stock.
Yeah,
Catherine's doing serious research on.
Okay, do you want to share your screen? Shall I unshare?
I can share my screen and show some stuff. So I have 3 examples that I've put together. One is I received some new samples that I want to QC in the lab. And so I have a script that I have that's a template that I can run to set up a series of experiments that will run in the lab to check the quality of those samples.
Sharing your screen, and then
yes, I can do that. Let's look at that and share. Okay, and then my other 2 examples, I have some data that I can walk you through a little bit of analysis that I've done on some fluorescence kinetics experiment. And then I need to repeat a fluorescence kinetics experiment this week, changing some of the parameters about mixing that I'm interested in. So is any of those 3 appealing to start with, or?
Let's do the one.
The fluorescence kinetics is the model system I was trying to pitch you on in that email that we could use to show.
Okay, let's see that. That would be great.
So the data analysis or running of the experiment?
Well, do both. Show the command that was used to run the experiment, then the data you got and the analysis and the fitting you do.
Sure. So in this experiment, what I'm trying to do, so normally when we're doing a fluorescence kinetics experiment, we're mixing together a duplex of DNA that has a fluorophore and quencher. And so when that duplex is duplexed, the fluorophore is quenched. And then we're adding a releasing strand that binds to that duplex, releasing the fluorescent strand, and we see fluorescence. So we just stick that in a plate reader, we read at the appropriate wavelength, we inject our releaser strand, and we see an increase in fluorescence. So in this case, I used a script, and I have some variables that I set up. I'm mixing
this is gonna get
some
fun and complicated fast if we're scripting it already.
yeah. I mean, this is a experiment I've already run. So but I have my water, my favorite buffer, PBS, which is actually the one that you just enqueued or will enqueue in the lab.
Yeah, you're
to
about
have
to make a
liter
liter of this buffer. That's
my favorite buffer. I have my DNA oligo samples, and I have a bunch of different concentrations that I'm testing. So without getting into too much detail, the basic idea here is I'm going to do a sample manipulation to mix all of my samples together in a plate. I'm then going to incubate that plate at 95 degrees to prepare the samples, to break the DNA apart. To
Can you scroll down a bit and let's see?
Oh, yes. So here's a bunch of setup stuff. Math to do some dilutions, explicitly specifying which wells samples are going to go into. And if we scroll down a little bit further, here's my sample manipulation call itself, or the operations that are going to happen. So defining some samples, saying like, let's scroll down to actual transfers down here. So I'm mixing
this
some
is
things.
not an uncomplicated experiment.
It's a very complicated thing. Sorry.
Well, just think what you'd have to do if you didn't have Emerald to do it.
Yeah, I'd have pages and pages of scratch notes and check marks after each one, so I make sure I'm pipetting into the right wells of every plate.
How long would it take you? I'm just curious to give me a sense of this. I mean, like if you were doing this experiment by hand, how long would it actually take to do it? Is this like a week long? Follow a day?
No, this is a relatively quick one. So to do 3 steps, setting up the plates, incubating them, and then reading them in the plate reader. So step 1, setting up the plates would probably take me a couple of hours. The robot can do it faster because it can pipette more things at once. I can only pipette one thing at a time. The robot can do 8 or 96.
Well, and here's a really impressive part, right? So like, yeah, it's like probably the better part of half a day at work, right?
Yes, yes.
But also, how many experiments does your team have running right now? If you flip over to your tab, Here's the cool part.
We are maxing out our threads.
12.
So we're using all 12 threads, and we have an additional 14 experiments waiting to begin when those threads are.
And that's a team of how many? 3?
Yes.
Yeah, there's 3 of you doing that. So that's the cool part too, because it just like spinning up servers, Catherine can spin up as many experiments as she can dream up here, up to the size of her account. So she can run 12 simultaneously. That's like what the account's provisioned for. And the rest will sit in queue. And as soon as any one of those 12 finish, it will roll on automatically to whatever's in her backlog of the queue there. So
yeah, it's amazing. I mean, the effective increase in productivity seems fairly spectacular here. Because I mean, otherwise would you be having 12 people to do, one person per thread if you were doing this by hand?
Oh, at least, because this runs 24/7. People would have to go home at the end of the night. So.
Okay, so that's 36 people or something with 3 shifts or whatever.
3 shifts, yeah. Yeah, it's probably a bit of an exaggeration because you're waiting for like reactions to happen and there's dead time in the middle of that. But pretty much, if you needed to keep the thing running at constant velocity, you would need to have somebody on staff at all points. And even the communication barrier would be a nightmare because in a traditional laboratory, Catherine wouldn't have computational language to specify what she's doing. So the ability to hand it off to somebody else would take, weeks of training before she could even give them the experiment and continue it when she's not there.
Right. It's very impressive. I'm also, unfortunately, I'm watching the time because I have to do something completely different at the top of the hour, and but this is exciting. So
what we can set you up here because you have an account now, right? So
yeah. But I want to get some sense. I mean, this is very interesting to see, and I don't know, chemists who are here, can you any comments on what you're seeing or questions?
Well, I did something similar to this ages ago with an old Brooker NMR, and I had to program it with a paper tape to run a series of experiments overnight, changing various decoupling frequencies and so on. And it took me forever to get that tape written and get the experiment to run beginning to end without failing for one reason or another. I mean, this automation is spectacular.
Yeah, this is a whole lot more impressive than the Zymark robotic system I helped build at in a pharmaceutical company years ago.
I've never built anything like this, but I'm still also impressed.
It's a composition of a lot of those machines too. So even like Robert, that NMR, it's not using paper tape anymore, but there's a big 500 by Bruker, probably even the same manufacturer, that has actually that instrument isn't so terrible. It has something close to an API we can talk to connect at the end of the day.
Yeah.
But it's all the like sample prep, and then there's 200 plus instruments that are networked together in this environment. Try to make a whole laboratory at the end of the day, which is the exciting part. And then everybody can be remote, doesn't ever have to set foot in it. So you can see why we're excited about every time you guys add features. Like, I mean, we're just like so excited when we could plan the whole reaction network. That's basically what you do in total synthesis. Is one of the two things is sit down and plan out what reactions you're going to run, and then two, physically carry them out in the lab. And if you could do all of that in a notebook, and
right,
that's straight up science fiction. That's awesome.
Yeah. So how close are you to actually, I mean, could I take Woodward synthesis of strychnine, for example, and run that?
So the biggest limitation right now on reactivity is we're only doing low energy stuff at the moment. So if you do like bioconjugations and that sort of thing that you could do in a liquid handler, it would be happy. There's a bunch of bigger, more serious reactors sitting in the corner of the lab right now that are on our to-do list to bring online. So I said there's 200 instruments online. There's another 45 we're still working on. And those are the ones that are like big jacketed reactors that we could run high energy chemistry in that we're going to bring online. So right now you have to camp it to low energy stuff. Part of the reason for that is just we haven't gotten to it yet with all the other stuff that we're doing. And the other reason for that is I'm sort of avoiding it because we want to do like if these reactions are happening to create these novel compounds, from a safety perspective, we're going to have to work it out such that you have to run a very small version of the reaction before scaling it up.
Mm-hmm.
Right. So we know the calorimetry, we know the pressure generating that. And, if you create a problem, you do it in a tiny vessel that just goes poof instead of burning my whole building down at 20 liters reactor. Yeah. Right. Yeah. So that system, the sort of reaction testing system where we'll force you to run text reactions is something that we have to bring on. That's kind of a framework before we do the higher energy reactivity right now.
Sure. Yeah, that makes sense. Can you do fermentations?
So we can do everything in vitro right now, and especially under the analytical heading and under the bioorganic heading. So all the biochemistry stuff, if you cut back to the website, you can see, actually, maybe I should share instead of Catherine.
Yeah.
Okay. So why is it? Okay, here we go. Instrumentation. So most of what we have online is very heavy in the analytical space, which to your project may be very helpful because there's a bunch of observables here. Mm-hmm. There's a ton of spectroscopy. Every different manner of spectroscopy is pretty much online. Tons of different readers for this. There's a lot of mass spec too, and LCMS. So in particular, if you're going to do super reactions, LCMS is probably the easiest way to sort out what's actually in there at the end of the day. GCMS as well, if we get smaller in size. And then from a bioassay capability, it's things like ELISA and qPCR and Western blot, DNA sequencing. BLI is that's for measuring interaction. So if you want KEQs and K-ons, K-offs, you get that from that. DSC, same thing, is for getting interaction energy, UV melting, that sort of thing. So it's all very in vitro right now. The stuff that's on the to-do list is really twofold. One is the cell culturing. So right now we only deal with cells after they're dead, but in the next couple of months we're bringing online the cell culturing system for the first time. So that'll allow you to do, live cells in a batch, and all the stuff you need for microbiology too, like colony picking and spread plating, maintaining your cells with cell splitting and all that. That actually happens on a giant robot. This thing here, for doing that sort of thing. That's what's being worked on like today, presently. So you'll see in the next 2 quarters ish, these things will start to release. And then the thing that's on my backlog that we haven't started yet, that you would want for the high energy chemistry. These are the big reactors. So the milligram scale reaction, the gram scale reactors, the big reactor reactors.
Yeah.
And then there's some exotic ones too, like flow synthesis and microwave and electrochemical. Part of what's nice about this is I can put a bunch of exotic equipment in there that's rare use. And because everybody's sharing it, it makes sense to have that around. So if you have a need for something weird, that's already true in like the spectroscopy space. So not a lot of people do what's something weird. CD is extremely weird. But we have this equipment online because if everyone's sharing it, this makes total sense to have available to everybody.
It's very impressive, and I really want to learn it because I mean, what do you think that the learning curve for how much do I have to understand about sort of the details of how these experiments work? This is kind of like if I'm going to do an integral, do I have to understand how things work, or can I just type it in? What given that I know,
a very interesting question. Everything usually comes from the other direction. I take a bunch of chemists and biologists, and they're like, "How long is it going to take for me to learn Mathematica so I can tell it how to do stuff in the lab?" I don't know if anyone's ever commented from the other direction of.
Well, but that's what you're going to be able to enable.
Yeah, I mean, absolutely the blurring of the lines. What is computational anymore becomes a weird concept.
Right, but my question would be, so there's sort of the question of what the mental model of what's going on is, and to what extent I have to be. I think we have to do some examples here.
That's why I think if we set you up with model systems, like there's a couple of Catherine, I'm thinking about the total mediated strand displacement reaction with the dual dye, so you can see binding and displacement as two separate steps. If we do the ugliness of synthesis and QC of that up front, Stephen doesn't have to worry about, like, that's just making sure the compounds are pure. We have exactly the thing we want to play with, and then show you how to run the actual reactions so you can monitor them and see the kinetics coming out.
That'd be great.
As you change species and as you change concentrations and stuff like that. That's really where the heart of the interesting math would come in anyway, to be like, well, how do I model this system? And we'll show you how, if you try to fit, you throw it into, there's an analyze kinetics function. And Catherine, you have an example of that set up, right?
Yes, I do.
Where when things work nicely, it's beautiful because you can extract rate constants. And when you get these model systems that just break everything, it melts down and nothing fits.
Yeah, I'm happy to share.
Yeah, could you just share for a second, and then I probably should run along to my quite different meeting here, but
so I don't have the perfect example, but I have a realistic one, and this is this experiment that I was describing earlier, where I was piloting a new version of doing this experiment. So here's some raw data. Here's some fluorescence. We see fluorescence over time. Lots of different examples of that.
Curves there.
Here's my actual interesting reaction. I've added my releaser. I see an increase in fluorescence.
Hold on. So this is over the course of many hours.
Correct.
This is so that some reaction is slowly happening that is changing the amount of fluorescence in your sample.
Correct.
Okay. All right. I'm just trying to, yes. Okay. Please go on. Yeah.
Yeah, I have a let's get you the better picture of that, where I've joined the baseline. I do an injection on the robot, and then I read the plate again to see the increase in fluorescence. So here you can actually see that there's very little fluorescence when we initially start. I add in my releaser in this blank area, and up goes the values, right?
Okay.
So then we scroll down to the bottom, and here I've used a standard curve to convert to concentration instead of fluorescence.
Okay.
I have a very simple mechanism that I'm using to model this. Just my reporter plus a releaser gives me fluorescence, and sometimes it gives me garbage. Just a very simple.
That's the reaction diagram like that you were drawing before. And there's two rates, KFF1, KBB1. That's forward one and back one. Is that what that's supposed to be?
Yes.
And K forward, K back 2 are those are the rate constants that we would need to fit to understand empirically what the rate of this reaction is, right?
Right.
Mm-hmm.
So I'm using the analyze kinetics function. I'm giving it just a pile of information saying these are the data that came out of it. Here's when I did the injections. Here's much the concentration information. And here's some starter rates that are in the ballpark of where I think this should end up being. And it spits out an object that I can then plot and compare.
You can actually see from the error message just running fine minimum at the end of the day.
Yeah.
To get the thing.
And it didn't come out perfectly because I had, this, you could see my maximum concentration was supposed to be 50 nanomolar, but with my standard curve conversion, things are a little off. And this is a piloting experiment, so I didn't expect it to be perfect. So we're going to iterate and do better next time. But you can see the like dotted, non-joined plot lines are my actual data, and the solid lines are the fitted data based on the rates that my analysis came up with. So we're pretty good at the lower concentrations that I injected, and as we get higher and higher, the standard curve issues become apparent. Yeah, so that's an example of just a quick example of doing an experiment in the lab using some Mathematica tools to do.
Well, now you can simulate it too. So like, do you want to run a simulation with those rates swapped in so you can see what happens when you change concentration?
Yes. I have not done that in a while though, and I don't think I could do it well on the fly.
For another time.
Right. Yeah. Okay. But I mean, so what happened here is you started this experiment, it ran for a few hours, then you got the data back, you fitted it, you have this way of essentially trying to fit the parameters of a differential equation to the data.
Yes.
Which seems extremely similar to what one does. I mean, I'm just trying to think whether we even have capabilities. I think our control systems functionality has some capabilities for fitting differential equation parameters, in effect. I mean, it's not usually used this way, but I mean, that's interesting in its own right to understand, are there fancier, I mean, it seems like you've got a perfectly good way of fitting the parameters of the differential equation. But then.
Right, and this is a well-behaved system, so that the models will fit. We have to work out obviously whatever's going on with that standard curve, not actually topping at 50. But the models will fit and we'll get nice rate constants. And the reason we're doing this is once we have the rate constants, you can then simulate the behavior of, okay, now I take these molecules and put them in a different context, different concentration with different other reactions present, since I understand, that empirical rate constant, those K values, and predict what their behavior will be in that system at the end of the day. That's why we're drawing rates out of this. Is Catherine makes larger and larger, more complicated systems. So this is one of the baseline components that will end up in those larger systems.
We do something very similar in solutions. One of our clients with epidemiology models and trying to predict if we vaccinate this age group or that age group, what's going to happen.
Let's see. My computer has, but somehow between its virtual machine features and everything else, my computer has now become basically unusable. So I can watch the screen but do very little else. Very interesting stuff, and I'm not able to.
Yeah. So what we could set you up with here is, and what we should do is probably give you like one well-behaved system first, where it's like, this is what it normally looks like when the kinetics is fine. And we can do like this, like the example Catherine is doing here, where it'll work. We'll get numbers out of these. We'll be able to fit them. We'll be able to predict them. And then I can give you the sort of the model breaking one that we came across where nothing seems to fit. And that would be really interesting to sit down and play with different math on how you might model the system without having to go all the way down to the level of molecular mechanics, still thinking statistically. But like you said, caring about the states of the molecules that are not just the present concentration of the species, but the evolution of the concentration of the species might be
right.
Yeah.
Yeah. Right. Very, very great stuff. I should go, but thank you to everybody here. And I wonder if that will make me. No, I still don't have. Oh yes, I've got my mouse back. Oh, how exciting! Well, great. To be continued. I mean, this is a good start. I realize I have a huge amount to learn here. And I would be, it would be great to get the initial chemicals plus tutorial, so to speak. Sounds like the next thing. And so when you prepare these chemicals and they just normally I'm used to software, so it's just like you have the data sitting on a disk somewhere. But in this case, if you prepare the chemicals, they have to be sitting in some physical test tube somewhere, and then they last for some amount of time, presumably. Or are they like data on a disk where they last forever?
Different chemicals can last different periods of time. The model system we're talking about, you keep it stored in a condition where it'll last several months, up to maybe a year or so before it starts to degrade.
All right,
but
okay.
the ugliness that we'll try to do it for you up front, which I was sort of describing, is these are components you can kind of commercially order, but then you have to quality control them because sometimes they'll be the right thing and sometimes they won't, and their purities will be all over the places, so you might have to purify them. But we can do sort of that grinding chemistry up front to get you to the interesting model system and then let you play from that point forward if you want.
That'd be great. Yeah, it'd be great. All right, this very impressive stuff. And by the way, for the Emerald team, it's just, spectacular. I have a bunch of detailed comments about UI kinds of things and so on, which I'd be happy to.
I want to get into that when you have the time. Absolutely.
Yeah, right. But what an impressive thing. Look forward to actually having a chance to use it. All right, we should run off here. Thanks very much to everybody, and see you later.